Well, it's just about 7:30, and I see people are walking into the room or coming online here. But I just wanted to welcome all, and Dr. Vega will be doing the formal introduction for Dr. Ganesh Rao. But as all of you know, this is part of our virtual Grand Round series, which has been going extremely well. And next month on November 25th, we will have Dr. Bernard Bendock with us. But to introduce Dr. Rao, who's actually well known by all, Dr. Vega is here. So I will mute myself and let him take the microphone.
Thank you so much, and welcome, ladies and gentlemen, to our BIDMC Virtual Grand Rounds. We're privileged today to have Dr. Ganesh Rao from Baylor College of Medicine from Houston, Texas, who has done amazing work over the last 15 years at MD Anderson before being named the chair at Baylor. And he comes today with a very important topic on modeling malignant progression in glioma. Prior to that, he did serve as the CNS president, also has won numerous awards from the AANS and CNS, including the Press Award for Excellence in Basic Neurological Research. And so it's my honor to present my mentor and a very good friend as well, Ganesh.
Thank you, Raphael, for that kind introduction. Let me just make sure I can share this properly. Can you guys see this now?
Yeah.
Great. So it's a tremendous honor to be asked to deliver Grand Rounds, even virtually here at Beth Israel Deaconess Medical Center. I'm really grateful to all of you for the invite, and particularly Dr. Ogilvie and Dr. Vega for inviting me. I actually realized in putting this talk together how familiar I am with your outstanding program and faculty. I've known Dr. Stippler for many years, actually. When I finished my term as CNS president over a year ago, I had the good fortune of working with Martina for the last several years. She is an absolute star and a tremendous asset to organized neurosurgery. I'm really glad I've gotten to know her. And as Raphael mentioned, when I was faculty, I worked with him over the last, I think he was a fellow my second to last year there. We even got out in the basketball court a couple of times, and you'll be surprised to know he moves pretty well for a guy of his size, so I was impressed. But I'm quite proud of you, Raphael, for all you've done since you've been there. I've heard nothing but great things about your practice there, and so congratulations to you on a great appointment at a great center. And I'd also like to acknowledge Drs. Alterman and Ogilvie, whose careers I've followed for many years, and I'm certainly grateful to them for the kind invitation. It's a real honor. So this is just a brief outline of my talk. I'd like to present some work from our lab that was recently published. My main laboratory interest is in low-grade glioma. I've focused on malignant progression of these tumors from low to high grade with an interest in both the genetic or molecular drivers of progression and more recently some of the immunological drivers. So there's a few connected stories that I'll go into here. Personally, I think these are some of the most challenging intraaxial tumors we deal with in oncology, not necessarily because the surgery is very difficult, but actually because the management has evolved over the last several years. And it's evolved since I was a resident. It was not uncommon to view these tumors as benign at best, or indolent at worst, that they would sit there for years and not really do anything. And we would frequently tell patients that these tumors were nothing to worry about, and we wouldn't even always follow them. But as molecular markers became apparent, primarily for prognostic purposes, for example, the 1p19q co-deletion, we would occasionally biopsy them. And then, as you know, just over a decade ago, the IDH mutation was really was described, and more recently, we've embraced resection for these tumors. And I'll briefly go through the data you're all familiar with in some of these studies, and then the relatively recent studies like RTOG 9802, for example. But I think it's pretty clear, again, from the data that observation is no longer an option. We still get referrals in which a patient's been told by a neuro-oncologist or an oncologist that maybe doesn't know the data that this is really a benign condition. And we know, thanks in large part to Hughes Defoe from Montpellier, France, that these tumors essentially always grow. It's really just a matter of time. So if you look at the x-axis on these graphs, you can see that we're talking about long periods of time. A lot of times these patients come to your clinic. You may scan them a couple of times and they move on. You lose track, but we know now that these tumors will grow. Retrospective studies, this one from the Hopkins group, really showed that extensive resection, this is really one of the first few papers I recall, again, over a decade ago, showing that the extent of resection was really associated with improved survival for low-grade gliomas and also decreased the likelihood of malignant progression, which is what I'm interested in primarily. The definitive data really supporting extensive resection came from Scandinavia, in which resection conferred a significant survival advantage when compared to biopsy. I think many of us are familiar with this data now. It's been, kind of all over the place in terms of, being presented. And this is sort of regardless of the histological or molecular subtype of the tumor. People sort of focus on the survival data here, but I'd also point out that they did report the malignant transformation rates, and in fact pointed out that the rates were significantly lower of these tumors turning into higher grade tumors if they were more aggressive in terms of their resection. And finally, kind of managing these tumors with radiation and PCV, procarbazine, CCNU, vincristine, is better than radiation alone. The key here is that patients undergoing a gross total resection in the low risk category, these are patients under 40, for example, may not need to be exposed to radiation or chemotherapy. So, for example, I just operated somebody who was 32, had a classic low-grade glioma, oligodendroglioma, histology, 1p19q code deleted, got what we call a gross total resection, and this patient is now essentially not going to need anything else because he's in the low-risk category. And the survival data, I think, really bears out the fact that these patients will do well. At our place, we generally test patients over 40 who've had a gross total resection, or treat patients, rather, I should say, with PCV, and RT or temozolomide, depending on the patient or the neuro-oncologist. For a patient who's 41, I think that's, for example, a case where we might have some controversy in terms of should we commit them to radiation and chemotherapy. I tend to favor watching those patients. Hughes Defoe, for example, has advocated holding radiation and considering re-resection if there's a recurrence. So I think the jury's still out on some of these finer points of how we should manage these patients, but it's clear that over the last several years, the posture has changed from one of maybe watchful waiting or minimal procedures, for example, a biopsy, and shifting that more towards intervening with surgery. So here's a couple of examples of how these cases might go, and in fact, I think they illustrate still the complexities that are associated with these cases. Here's a relatively young person who has an IDH wild-type low-grade astrocytoma, who undergoes resection. We perform standard cortical and subcortical mappings indicated. The patient has a gross total resection. 7 years later, shows up with a small recurrence, which is resected, but now it's higher grade. But you can see the time period is quite different than what we normally expect from an IDH wild-type tumor. This patient has gone 7 years without a recurrence and then has a small one. But after surgery, 3 years later, he has a significant recurrence and succumbs to the disease. So again, this sort of defies what we'd normally expect of an IDH wild-type tumor. Here's a patient with an IDH mutant low-grade astrocytoma who undergoes a gross total resection. But then he develops a massive recurrence just a couple years later and succumbs to the disease. So this is one of those frustrating cases where you tell the family, oh, he's got a favorable molecular profile. He should do well, but sometimes these things defy us. And I think these cases sort of underscore how the molecular imaging and histological characteristics of these tumors remain somewhat poorly understood. Certainly the heterogeneity of glioma is better understood now due to molecular profiling. We know that this is really multiple diseases with distinct behaviors based on the molecular characteristics. There's an early split with respect to IDH status when you look at when we characterize these tumors, and then there's other molecular characteristics that really shape the tumor. So each of these has different clinical profiles, and although we test for some of these genetic features, some remain either obscure or not well characterized. In this group, in fact, our group contributed by identifying transcription factors that seem to play a role in progression from low to high-grade glioma, but these aren't things that are routinely tested for. A more recent study to which my group also contributed data looked at the longitudinal trajectories of glioma. And again, underscored the fact that heterogeneity is still somewhat poorly understood and even refuted some earlier findings, notably the hypermutator type tumors. So these are tumors that were characterized by Joe Costello's group at UCSF several years ago. Patients were getting temozolomide, were being identified later on as having a very aggressive kind of recurrent tumor that was labeled a hypermutator state. This paper looked at those in more detail, didn't find that necessarily there was a difference in survival. So again, things change over time. I think this is probably the strongest statement from the paper, which is that it really underscores the fact that the challenge of trying to treat these tumors, that the development of these tumors progression really behaves in a stochastic manner. We really don't quite understand what causes these tumors to progress. To confuse things even more is the nomenclature continues to change. This is probably going to come in the next WHO update. These IDH mutant tumors, which devolve to the higher grade variant, thus going from a high WHO grade 2 to WHO grade 4 tumor, are now really called astrocytoma IDH mutant grade 4, distinguishing them from glioblastoma. So I think what you're going to see is that if you know a patient had an IDH mutant tumor as a 2 and it progressed to a gnarly 4, you wouldn't call that a GBM. We're going to start probably calling that an IDH1. IDH mutant astrocytoma grade 4. And these tumors are all getting more and more better characterized by the molecular features, which is really the integrated pathology report that I'm sure you're all familiar with at your center. So I'm going to focus some research from my lab in which we've looked at some genetic drivers of malignant progression, and also later on talk about some immunological drivers. So we've been studying tumor samples from patients who've had surgery or biopsy for low-grade gliomas at MD Anderson, where I was before moving over to Baylor, that we know have progressed to high-grade tumors. So in some cases, we actually have matched samples from the same patient. These can be, separated by several years, sometimes decades, in terms of when they were originally biopsied to when they transformed. We've primarily been focused on RNA sequencing. That was sort of the molecular route that I went initially. There are some challenges with that. Obviously, a lot of these samples are from FFPE. They're severely degraded. We had to discard some of those with low mapping rates. But you have to find a good, what we call biostatistics person, and have them employ what I like to call informagic. So for those of you that are familiar with this field, there's a lot of hand waving sometimes that goes on, but you can get some very interesting data out of this if you've got high quality samples. So we've identified several candidate genes. So we've looked at differential expression, both log changes in expression, fold changes in expression, and just straight up, differences in differential gene expression between our low grade and high grade samples. And again, I've tried to identify those that seem to be more expressed in the high grade tumors compared to the low grade counterparts to see if these are driving this progression. And we've also looked at the literature to support some of the, looking at some of these genes. An example we recently looked at was survivin, or BRCA5, which is an inhibitor of apoptosis. This gene had actually been well described in glioblastoma in the early 2000s as having a negative influence on survival. And we wanted to know, could it actually influence malignant progression of low-grade gliomas? What we found is that it's somewhat complicated. It's not a simple gene. There's several splice variants of survivin that may play different roles. In the low-grade glioma data set from TCGA, we actually found that patients who have high expression of this gene in their tumors have a significant survival disadvantage compared to what we call non-expressors or low expressors. This is actually about a 5-year difference if you look at the x-axis compared to non-expressors. And when patients with low-grade gliomas succumb to their disease, it's usually because of malignant progression. So we believe that this poor survival rate is a surrogate for malignant progression. So a lot of these patients have variant 1. Remember from the earlier slide, variant 1 seems to be the dominant isoform of survivin. There are several other ones. Variant 2 expressors, if you see here, survival is actually worse, 380 days on average versus 482 days for the variant 1 expressors. Again, this is in GBM cases, not in low-grade cases. But we wanted to know what the functional consequence of expression of these different variants was in vivo to see how they contribute to malignant progression. Now, we do this by using a genetically engineered mouse model of glioma based on the RCAS NTVA system. This allows us to model ectopic gene expression from glioneuronal precursors, and we can model ectopic gene expression in a cell type specific manner, and I'll go into this in a little more detail. It's actually a pretty commonly used model, but there's some nuances to this. So it allows for postnatal gene transfer in a cell type specific manner. We use a viral vector that is basically a retrovirus in which a gene of interest can be inserted. the mice are transgenic for the receptor for this vector, which is essentially a modified chicken virus. So the vector is actually replication competent in chicken cells, but not in the mammalian cells. And then the host genome actually takes up the virus and integrates the gene for expression. What's very valuable about this model is that the mice are immunocompetent and possess largely the same types of immune cells that dominate the microenvironment in humans. And I'll show you why that's important later. Here's the model graphically. So expression of the TVA receptor is restricted to cells that are expressing nestin. So again, glioneuronal precursors, only those cell types will be able to incorporate the retrovirus. The retrovirus is actually quite simple. It's a typical retrovirus with your typical retroviral genes. Your gene of interest can be inserted with very easy cloning techniques. And then once the virus is taken up by these cells, the retroviral genes are spliced out. And then the LTR is a strong promoter, and it basically continues to transcribe this gene. So these cells are now, in some cases, secreting this product. In other cases, they're just keeping it intracellularly and exposing the cell to the gene of interest. So basically, we created survivin variant 1 and 2 genes and express these in our PDGF-driven tumors. So this model, like many models, requires sort of an initiator gene to induce tumors. It's been well described that activation of the PDGF pathway actually can drive or can initiate tumors that have a low-grade phenotype primarily. So that's the dominant phenotype in this model, that we see a low-grade kind of variant of these tumors. It's quite reminiscent of what we see in human patients, kind of hypercellular tumors that are very bland. There are no high-grade features like microvascular proliferation or necrosis, but you can combine RCAS vectors to see how they synergize or contribute to each other, cooperate rather, with PDGF to drive a tumor formation. And in fact, when we express variants 1 and 2, we see that the co-expression of variant 2 is much more likely to drive a high-grade tumor histology. And this is characterized by these high-grade features that I alluded to, including microvascular proliferation and necrosis seen here. This is a PDGF plus variant 1 tumor where it's quite bland. Again, you see the dominant phenotype here is these low-grade tumors. And here I'm just demonstrating the fact that we can actually then stain for variant 1 and 2 in these tumors to make sure or verify that they're being suppressed. Unsurprisingly, survival is also considerably poorer in mice in which we co-express PDGF and survivin variant 2 compared to variant 1. In fact, survival with variant 1 is similar to what we see with PDGF expression alone. So it seems that although variant 1 is the dominant form that we see, it doesn't seem to have this striking effect in causing malignant progression in patients with or in our low-grade model. Variant 2 seems to, but again, it's not the common one. It's only seen again a minority of patients. Both genes are anti-apoptotic, which you would expect. That was well known in the literature. We characterize this by looking at the apoptotic index by CC3 and cleaved caspase 7 expression. We did see a difference in the extent of angiogenesis as measured by CD31 expression. That's quantified here. We also did microvessel counts and density. We actually discovered that variant 2 is actually highly expressed in endothelial cells. This is a known feature of this variant. And we demonstrated that variant 2 can promote endothelial tube formation, though less frequently expressed. Variant 2 seems to have more of an influence on the malignant progression of gliomas. We were sort of gratified to find out that this has been looked at in leukemia, that variant 2 seemed to be restricted to endothelial cells. So it was starting to kind of coalesce around this pro-angiogenic story with respect to this particular variant. So nice kind of story there showing how we can identify these genes by gene expression between the samples, and how we can functionally validate them in a mouse model to show that we think they actually do what they think they do. But it doesn't always work out this cleanly, and I can tell you that we sort of looked at this list and thought it was a gold mine of ways or genes that we could look at to see, are these contributing to malignant progression. FOXM1, for example, had been identified by other groups as being highly expressed in high-grade tumors compared to low grades. Murat Ganel's group at Yale had reported this several years ago, and it was a great candidate. It had a highly significant fold change in expression, and so we thought, oh, surely this will be one. But when we modeled this, and even a constitutively active version of FOXM1, which is truncated to allow constitutive expression, we were unable to show a survival difference or even the incidence of high-grade tumor formation compared to PDGF expression alone. And I think this really highlights the importance of functionally validating these genes with more than just, kind of an in silico approach, that we really need to prove that they do something. And in fact, again, this gene had been identified many years earlier, and it's a very complicated gene that interacts with other complexes, and so we're actually now looking at this not in isolation, but in the context of some of these other genes that it interplays with. And we're also taking, I think, a more systematic approach to this by identifying genes that correlate with poor survival and finding the intersection of those genes that are differentially expressed, and then narrowing that down even further with master regulator motifs, and using really the cases where there's much poor survival to help us identify genes that really drive malignant progression. So as I mentioned earlier, I do think that these patients that succumb to their disease early are progressing more rapidly, and so we've actually sort of split our cohort of patients even further into kind of poor survivors versus some patients who do better, to really just kind of dial down that signal to help us identify these. So we're in the middle of this now, and I'm hoping that we'll have some results soon. We're sort of at the mercy of our bioinformatics folks to help us identify these and then functionally validate them. So we're looking at other ways that the tumor microenvironments can influence malignant progression in terms of chemokine expression. Specifically, I'm going to focus on a story called CX3CL1, CX3CR1 signaling. This work was done in my lab, actually, by one of our really outstanding residents, Sung-Ho Lee, who had done some previous work in this area during his PhD. This particular chemokine signaling pathway is known to affect the microenvironment, so it ties in nicely to the next story. Although there's some conflicting data regarding its effect on tumor progression. So there's some data and literature suggesting that actually activation of this causes malignant progression, and some that says it actually is protective. What piqued my interest in that actually is that there's a well-described polymorphism of the receptor CX3CR1 that correlates with increased survival in GVM. And this particular polymorphism actually inhibits binding of the ligand CX3CL1 or fractalkine, for those of you that know it, to the receptor. This is a very clean chemokine signal ligand to receptor in terms of their cognate. So there's no other, like dirty signaling or other ligands that bind to CX3CR1. That's not true of all chemokines, but this is one of those rare ones where that is the case. So in our own data set of patients, again, who have progressed from low to high grade, and again, these are IDH mutant cases, as you would expect, we find a pretty striking survival difference in patients with the polymorphism. So here, the red line are patients with the polymorphism, either homozygous or heterozygous, and then these are the patients, the blue line are the patients with the wild type version. And you can see overall survival, this is years, is significantly different in terms of their survival. That's true for progression-free survival as well. Again, a difference in years. So you can see, again, 5-year difference is not trivial for these patients when we look at them. So we did some gene expression profiling of these, and revealing really 2 genes of interest. We looked at 2 glioma stem cell lines, one with the polymorphism. So this is courtesy of Fred Lang, who has a whole set of GSCs, and we actually identified one that had this polymorphism in it and one that didn't, and looked to see, we identified an increased expression of CCL2 in the wild type cell line. We also see increased expression of immunosuppressive macrophages and angiogenesis from the tumors of these specimens. So we actually identified the human tumors and were able to profile them. So naturally, we wanted to validate this in the mice in the low-grade samples. We modeled this in the mice and find that mice expressing the wild type receptor have a survival disadvantage. And when we express the ligand in the receptor, we see an increase in macrophage infiltration and in angiogenesis. So that's characterized here. This is IBA1 staining. This is the macrophages, and you can see a significant expression of those where we co-express both the ligand and the receptor, and increases in microvessel density seen here when we express the ligand and the receptor. Again, all in the context of PDGFB. we don't get tumors if we don't induce them first with PDGFB. So this just activating this signaling pathway, which we did, I'm not showing the data there, is negative. These mice won't form tumors. So here's sort of a nice summary of our findings regarding the effect of the polymorphism compared to the wild-type receptor. The wild-type receptor seems to promote M2 macrophage infiltration and angiogenesis. So those of you who know about sort of macrophage polarization, the M2 macrophages seem to be immunosuppressive versus the M1 macrophages that tend to be sort of anti-tumor or immunosupportive. We're now exploring this in more detail. Sort of the next step here is to identify inhibitors of this receptor. We actually have CX3CR1 knockout mice that we've crossed with our NTVA colony to see how the phenotype can be altered with complete abrogation of the signaling pathway. And I think this may actually be a potential pathway to target or to delay or mitigate the process of malignant progression. So this sort of segues into kind of my next section. So we've seen how these genetic variants can actually influence the tumor microenvironment as well. As I mentioned with the CX3CR1 story, we're seeing how macrophages can be affected by this. And it's been known for a long time how the tumor microenvironment of GBM, for example, is incredibly immunosuppressed. And so we've been exploring how the microenvironment can contribute to malignant progression of glioma using our mouse model. As I mentioned, GBM, or the glioma microenvironment, is uniquely immunosuppressed. It's largely immunosuppressed due to macrophages and regulatory T cells. And we've observed that in both human and murine samples as tumors become more malignant. So in human and mouse tumors, we've noticed that as tumors progress from low to high grade, there's a significant infiltration of these macrophages. These particular macrophages are stained for IBA1, which is a robust macrophage marker. It's really my favorite macrophage marker because it always works in the lab. There are several other ones that are a little more finicky. But as many of you know, in human tumors, so these are human samples here. These are matched samples, patients who had low-grade tumors first that progressed to high grade, and you can see that there's significant macrophage infiltration of these tumors. And then in our mouse model, this is a whole mount, and you can see that in the high-grade area, we have a lot of microvascular proliferation and necrosis, that there's also significant infiltration of macrophages. And for those of you that do this work, you know that when we take these tumors out, even up to 30 to 50% of the overall mass that we're taking out is actually macrophages. So this led us, this has sort of led me down this path of being more interested in immunology. And for those of us that do oncology, you know that we all sort of have becoming forced to become immunologists. This actually worked from many years ago. So this work I did with Amy Heimberger at MD Anderson. And we noticed early on when we were modeling actually co-expression of BCL2, an anti-apoptotic gene with PDGF. One of the things we noticed was that the macrophage infiltration or counts were much higher in the mice that died early from the tumors compared to ones that survived to sort of an endpoint. And so, this is something it was sort of correlative. We didn't know what to do with it, but we have been looking for genes that seem to kind of promote immunosuppression. And one of those that we've recently reported on is fibrinogen-like growth factor 2, or fibrinogen-like factor 2. So this is one of these genes that lies at the nexus of immunosuppression and malignant progression in glioma. Interestingly, FGL2 expression is usually confined to T cells, endothelial cells, and tumor cells. And expression is induced with the monocyte macrophage lineage. It's also critical to Treg function, which, as I mentioned, is a big immunosuppressive component well described in the microenvironment. So FGL2 really seems to lie at the center of a lot of microenvironmental factors that make glioma unique. My colleagues Amy Heimberger and Shu-Lin Lee, who's a pediatric immunologist at MD Anderson, had shown a significant increase in expression of FGL2 compared to low grades, which prompted us to see whether or not it played a role in malignant progression of low to high grade tumors. They were more interested in this in terms of its role in GBM, and have actually even created an antibody to see if it can be used to treat patients. We actually looked at this from a low-grade glioma standpoint. So we queried TCGA for survival in patients with increased expression of FGL2. In all glioma cases, we see a definite survival disadvantage, but that didn't seem to bear out when we looked at just at GBM. So patients with GBM, which you can see here, the high expressors of FGL2 don't seem to have the same survival disadvantage that they have if they're started out as low grades, which you can see here. And again, if you look at the x-axis, we're talking about several months difference in survival. So the survival difference here is again in terms of years, almost 5 years. And we profiled our set of matched progressed cases. So here's an example of a low-grade glioma, non-enhancing lesion, resected, but then several years later has a recurrence, and you can see this is FGL2 staining here, significant increase in FGL2 expression in the high-grade tumors. So we actually obviously took this to the mice to see if it did anything. In our FGL2, when we overexpress FGL2 in mice with PDGF, we see a significant survival disadvantage. So that's this line here. This is PDGF alone. FGL2 by itself does not seem to cause any kind of tumor. And again, not surprised by that. It's never been described as contributing to tumor formation, but clearly plays a role. And so we investigated this further. We looked at, so obviously the high-grade tumor formation rate is much higher when we co-express FGL2 with PDGF. We see a shift from what's called proneural to mesenchymal subtype, which again, for those of you that are familiar with this is in the continuum of glioblastoma, where the OLIG2 expressing tumors tend to be more proneural and tend to do better clinically than the mesenchymal subtype, which we can characterize by CD44 expression here in these samples. And then we looked at FOXP3, which is the Treg marker, and that seems to occur actually pretty early on. So FGL2 induces its influx really by about day 20 or so. We induce the tumors, and then we sacked a cohort of these mice at various time points and showed that the Treg infiltration doesn't happen early, but by day 20, we see a significant influx of these regulatory T cells, and we can quantify them. And we do have access to this FGL2 antibody. It's actually an anti-mouse antibody, and we're able to use it. So Dr. Shulin Lee created this antibody, and we're able to treat it. And so we treated the mice and compared that to IgG control. So you can see with the antibody, we have a significant survival advantage with this treatment compared to the IgG control. Now, to be sure, these mice aren't cured. They still have tumors, but they are much more likely to be low grade compared to high grade tumors that we saw with IgG treatment. They tend to actually not shift to the mesenchymal subtype, so they seem to have low, or rather high OLIG2 expression, low CD44 expression. They have a reduction in the CD44 FOXP3 population. And what really was interesting to us was this macrophage story. So we see a significant reduction in the number of macrophages, and they seem to be shifting away from the M2 phenotype back to the M1 phenotype, which suggests that we can actually influence the macrophage population here, and that'll become important in the next story. So we're still investigating its therapeutic effect by figuring out how it actually works. Presumably, it has its effect in the periphery and limits the migration of Tregs and peripheral macrophages polarized to the M2 phenotype, but we're really not sure. There is certainly some evidence, and the kind of standard teaching as I've come to learn is that these antibodies are way too large to get into the tumor microenvironment of glioma because of the blood-brain barrier. However, as many of you know, the blood-brain barrier is broken down with these tumors, and I'll show you some data later that suggests that some of these antibodies may actually get in to the tumor. We're also interested in combining this with checkpoint inhibitors to see if there's synergy between these antibodies, and that's work that's ongoing in our lab now. So as our interest in the microenvironment of glioma has evolved, we've really begun to take a closer look at how unique it is. And in particular, this is focused on CD8 T cells. These are the cytotoxic T cells that are thought to facilitate the therapeutic effect of checkpoint inhibitors. And it was previously reported by Andy Parsa many years ago that the extent of CD8 T cell infiltration also correlated positively with survival. And we wanted to know, how do these cells shape or influence the process of malignant progression? So, to get at this, we actually decided we would actually remove the T cells. So we actually created CD8 knockout mice and bred them with our NTVA mice, and really considered three hypotheses when we did this to see, to see what would happen. One of these was that, cells or mice without CD8 T cells, once we induced tumors, would undergo more rapid malignant progression and their survival would be poorer. Or that without CD8 T cells pruning tumors with immunologically susceptible populations, there would be a lower incidence of high-grade tumors and survival would be better because there's not this selective pressure for the tumor to become more malignant or have these genetic changes. And then we had to consider the possibility that getting rid of these CD8 T cells would have no effect whatsoever, right? That maybe they're not relevant at all. You can deplete CD8 T cells, but it's actually quite challenging. You have to treat them continuously with antibodies to knock out CD8 T cells. So we genetically altered them by eliminating the alpha chain of the CD8 glycoprotein and crossed them into the NTVA background using speed congenetics, and then created these CD8 knockout and wild-type NTVA mice. And then tumors were induced by combining PDGF and STAT3. So STAT3, we reported earlier, it's a transcription factor. It's been well described in glioblastoma, and when we combine that with PDGFB, we actually get very high tumor penetrance. 100% of the mice get tumors, and they are very, the continuum is shifted significantly to the high-grade phenotype. And we can actually modulate the amount of virus that we expose our mice to. And so that way we can actually see, we can actually look at almost in a way, see how a rapidly forming tumor versus a slow forming tumor can develop. And we wanted to look at that in the context of this lack of CD8 T cells. And so when we induce these tumors, we follow them for about 90 days or so. Usually by then, all the mice have tumors. And as if you recall from the survival curves, if there's going to be an effect, we'll see it pretty early. The median survival with a combination of PDGF and STAT3 is usually about 50 days or so. So we had to verify that we were not getting any CD8 alpha in the brains of these knockout mice, and that's depicted here with no expression of the CD8 gene. And then after injecting the mice with the different quote-unquote concentrations, as I mentioned, so this is a low concentration of viral vectors, and you can see regardless of the background, whether or not you had T cells or not, there's no difference in survival. That's true no matter how much of the PDGF and STAT3 we exposed the mice to. So that kind of surprised me because I thought for sure we would see some difference given the role of CD8 T cells in certainly other cancers. And we didn't see statistically significant difference in the higher low-grade tumor formation rates. So we sort of scratched our heads a little bit and wondered, are there problems here? Maybe we didn't really knock this out. We did verify a lack of T cells infiltrating into the tumors. So these are the knockout mice. The red fluorescence is the T cells, and we didn't see any in the knockout mice. We did see a compensatory increase in other immune cells in the periphery, including CD4 cells, B cells, natural killer cells in the spleen and blood, but not in the tumor itself. In fact, we did not see any CD4 cells even in the tumor. Again, we didn't see these cells in the tumor, but saw these expand peripherally in tumor-bearing animals in the CD8 knockout mice. So here you can see in spleen, we're seeing natural killer cells, we're seeing B cells, but in the tumor, it's pretty devoid of any real immune cells, which, again, expansion in the periphery, but really we're not seeing much at all in the tumor itself. Now, we did see an increased number of macrophages. Okay, sorry. Using FACS, we also did see myeloid cells with a dominant cell. So these are the myeloid cell expression in both the wild type and knockout backgrounds, but didn't see really, these are CD3 cells, these are natural killer cells. We see very low expression of these cells in the tumor. We do see PD-1 expression on the myeloid cells, and that becomes important later. But again, the other populations seem to be quite low. So again, as I mentioned in the previous slide, we did see an increase in the number of macrophages in the gliomas of CD8 knockout mice compared to the wild type counterparts. These macrophages seem to be polarized to the M1 phenotype based on TNF alpha expression. We didn't see this type of polarization in the non-tumor bearing control mice, implying that the tumors are also playing a role here in terms of what happens to macrophage polarization. Using immunofluorescence, we again see the higher incidence of macrophages in the CD8 knockout mice. And the macrophages in the CD8 wild-type mice are arginase positive, again suggesting this M2 polarization. You can see that with the yellow staining here, co-staining. So we wanted to know, okay, the T cells, maybe they're there, but in wild-type mice, but they're being suppressed by PD-1 activation, and they can't mount an anti-tumor response. So we treated them with a murine version of the PD-1 anti-PD-1 antibody to see if that might restore the anti-tumor effect of T cells against the tumor. As you all know, there's been conflicting reports of the efficacy of checkpoint inhibition against glioma. I thought it wasn't going to work. It certainly has been a home run for other cancers like melanoma and kidney cancer, lung cancer, et cetera. There's a small signal, as you probably know from work that was done at UCLA, showing that for neoadjuvant checkpoint inhibitors, there's been a survival advantage. But we wanted to see if maybe these cells were inhibited in the wild type mice. And so, and again, there's been, as an aside, there's been preclinical work showing that checkpoint inhibitors can work when they're sort of given in the context of other treatments. For example, this is work that was done by Mike Lim, now at Stanford, who showed that with either radiation or even other adjuvants, you can actually get immunotherapy to work in murine models. So much to our surprise, actually, we saw a significant survival benefit with anti-PD-1 antibody in both the wild type and knockout mice compared to IgG controls. And so, of course, we didn't expect it to work at all in the knockout mice. And there's no difference actually in survival in terms of the survival benefit between wild type and knockout mice. So it seems to be working equally well in both backgrounds. It was really unclear how the survival benefit was occurring, particularly in knockout mice, because they don't have T cells. But we felt confident that we've shown that other effector cell populations that potentially could benefit from PD-1 inhibition, the CD4 cells and the NK cells, for example, were absent. But examining these tumors more closely, we find that macrophages are PD-1 positive, which is also well known. Irv Weissman's group at Stanford has shown this. But we see a significant reduction in them in the treated mice compared to the controls, in which the macrophage population remains largely intact. So again, these are the PD-1 treated. You can see a significant reduction of the IgE1 macrophages, and in the IgG treated controls, there's still a ton of macrophages there, and they are largely PD-1 positive. We did some quick nanostring analysis on these tumors and found that the remaining macrophages in the treated mice demonstrate a shift towards the M1 profile. In other words, the RNA from these tumors treated with the anti-PD-1 antibody fit the M1 macrophage gene expression profile compared to the IgG treated mice. We characterize these macrophages further by trying to differentiate them in terms of whether or not they come from the periphery or a resident microglia. So TMEM119 is a marker for microglia. And we found a reduction in these cells in mice treated with PD-1 in both the knockout and wild-type mice. We think the therapeutic effect is being mediated by these macrophages, but we also noted that antibody-dependent cell-mediated toxicity, or ADCC, also may be playing a role. An ADCC assay on a murine microglial cell line that we studied showed a reduction in PD-1 expressing macrophages after exposure to the antibodies, so there may be a role there for this. I personally think there's actually a direct phagocytic effect from these macrophages that's also been described by Irv Weissman in the context of colon cancer. But there's some people who believe that you need to have these antigen presentation to these macrophages for that to happen. There's work by Betty Kim's group over at MD Anderson that also shows that direct phagocytosis of tumor cells is possible with activation. So we're trying to tease this out now. And so there's some future directions here. Obviously, we're very interested in better understanding the microenvironment of the glioma in the context of checkpoint inhibition, mostly to understand what cells are mediating the effect, the therapeutic effect. And as we all know, neoadjuvant treatment seems to have some kind of therapeutic effect, whether it's radiation beforehand or giving these PD-1 inhibitors prior to surgery, for example. And of course, that may be a next step here. A big question that always comes up is, are these antibodies even getting into the tumor microenvironment, or is this a peripherally mediated effect? We've actually done some work on this and have shown, we actually labeled these antibodies, we absorbed them with a fluorescent marker, and in two different models, so in GL261 and in our NTVA mice, we actually were able to show that we do identify these antibodies in the tumor microenvironment. At least we're seeing fluorescence in a place where the tumor is, suggesting that it is in fact getting in. And whether or not it's probably due to disruption of the blood-brain barrier. But again, we're trying to characterize this better. So to summarize, again, my lab is focused on low to high-grade glioma progression. We've identified or investigated differential expression of genes between these matched samples, which have really been useful to us to identify these transcription factors that promote malignant progression in glioma. Both BRCA5 and FOXM1 are examples of genes that can be functionally validated in a good genetically engineered mouse model. And you can see that in both those stories, one clearly had an effect, although it was sort of the transcription variant that was having the effect, versus one that didn't work at all with respect to FOXM1. We've also shown how some of these genetic drivers for the chemokine story, for example, can modulate the tumor microenvironment. I do think that the tumor microenvironment is still may offer a therapeutic option for these patients certainly to mitigate malignant transformation. Certainly our results with FGL2 and even anti-PD-1 suggest a therapeutic opportunity. The question is, when would you give it? And that's something we're obviously struggling with. These patients can live a long time. You certainly would not commit them to some kind of treatment over decades, particularly in the context of immunotherapy, which can have some side effects long term. I think that if there's a role for checkpoint inhibition, if we start to see these tumors change over time, it'll need to be in the context of another therapeutic strategy. I think in isolation, it probably won't work. And if I can leave the residents with anything, it's when you see a low-grade glioma in clinic or wherever you do see them, you should really be thinking about how can we use surgery to help these patients. I think it's quite clear now that surgery has a significant role to play in treating these tumors, and it's one of those places where we can really make a difference. So got a lot of people to thank for this. My lab, Amy Heimberger's lab, who has really been very helpful, and particularly helping me understand immunology. Eric Solman, who's now at NYU, and then my funding sources. And with that, I am happy to take questions. I'm going to stop sharing, I think, if I can figure this out. Maybe not.
That's okay. Dr. Rao, thank you.
Sure.
For that amazing talk, and really trying to focus in on the importance of the cellular aspect of the microenvironment. This disease process, as we know, is more of a cellular disease, but that we deal with in a macro scale. Just out of curiosity, what's your thoughts on different therapeutic approaches versus, like, you talked about variant 2, and you did spend a nice time talking about like the PD-1 and the antibody part, but what type of genetic strategy or gene therapy would be implemented for something like this?
Yeah, it's a good question. I mean, going back many years, people may remember, even Fred Lang many years ago in a previous life, he tried to reintroduce P53, for example, into these tumors to try to get them to restore function. It didn't work. I think there's a role for that. The problem is that once everybody has their favorite gene, and we've all seen how targeting a single pathway just doesn't work, right? There's just too many escape mechanisms, particularly in GBM. So my sense is that for those of us that have been around long enough, I'm sort of a skeptic there. I don't know that this is going to be a way to do it, in terms of identifying a single gene or genetic pathway to do it. People have bashed, PI3 kinase, for example, for years, trying to get, suppress AKT, what have you, and go on, and it just really hasn't worked. So there's so many escape mechanisms. And we can also talk, we talk about bevacizumab, right? I mean, obviously that was supposed to be a home run and blocked angiogenesis. Well, it turns out, of course, there's 12 different ways that a tumor can recruit blood vessels. So in terms of therapeutic options, I think all of us are being more convinced that it's going to be a combination of things, and frankly, identifying maybe even, such a heterogeneous disease. We have BRAF mutations in glioblastoma, for example, that account for maybe 1% of these tumors. But I've seen, if you've got that mutation, you get a BRAF inhibitor, the results are actually pretty remarkable. So it may be that we're going to have to identify, everybody's tumor is so unique, we're going to have to create these very, very tailored treatments for people to try to get them to respond. The nice thing about immunotherapy for GBM, if it ever works, is that it's almost agnostic to the heterogeneity, right? So if you have a highly immunogenic tumor, and people are trying to characterize tumors now by their immune profile, that you can maybe predict with a biomarker who's going to respond better to checkpoint inhibition versus those who aren't.
Yeah.
Hi, Dr. Rao. This is Aristoteles, one of the residents. Great talk.
Thank you.
I have two questions. Question number one, has anyone studied if there is a difference between molecular profiling of these tumors on the periphery, like close to the transitional border, like normal brain to tumor, versus core of tumor? And question number two, are there any studies that they show an evolution of the molecular profiling after chemo and radiation? So you have the tumor, you take it out, you do chemo and radiation, and if there is any change on this molecular profiling immediately after this?
Yeah, to answer your first question, yes. There's actually been quite a few studies on this. There's a paper in PNAS from, I think, 2016, where they did targeted biopsies of the core of the tumor, the periphery of the tumor. There are significant differences in the molecular profile of the tumor, depending on spatially where it is relative to the rest of the brain. And even Amy Heimberger, again, my colleague at MD Anderson, has what's called a topographical atlas, where she's done multiplex immune staining, immunohistochemistry, showing significant differences depending on where you are in the infiltrating edge of the tumor versus the core of the tumor. There's no question there's a lot of heterogeneity, intratumoral heterogeneity there. And then respect to your second question, yes, there's a lot of, the paper I showed you, the longitudinal study of glioma progression, takes into account the changes, the molecular changes after chemotherapy and radiation. Again, that's that hypermutator phenotype that Joe Costello described, I think in Science 4 or 5 years ago, showing that there was a big difference, a big change in kind of the mutational load of these tumors after chemotherapy and radiation.
Dr. Rao, thank you very much for the talk. This is Dr. Eric Wong. I'm a neuro-oncologist. I have a number of questions. I remember when I was doing a sub-I at Methodist Hospital, I wandered into Fondren 12. It was a special place. Is it still a special place there? That's my first question. My second question, yeah, go ahead.
I don't know, actually. So,
okay.
Many years ago, about 15 years ago, Baylor and Methodist had a very ugly divorce.
Oh, I see. Okay.
And so, Methodist has its own program now. I've actually never set foot in Methodist Hospital.
Oh, I see. Okay. I have two scientific questions. One is that neurosurgeons do supramaximal resection, and supramaximal resection has a bigger role for low-grade glioma. And I would like to hear your philosophy of whether or not you're a believer in supramaximal resection, or even go with one step beyond. And I would like to know, because you have no marker to tell you where to stop. When do you stop? My second question is, you did your experiments on survivin and FCL2 on the PDGF background. Have you tried looking into the effect on an EGF background?
Yeah, that's a great, I'll take that second one first. I have not. The EGFR activation, there is actually an RCAS model that uses EGF, where you actually overexpress EGF. The problem is the tumor formation rate is pretty low. So it's not at least in our model, it's not a practical way to actually, you have to actually, a lot of mice aren't going to get tumors, or it takes a long time for them to form. So I haven't done that particular, use that particular background. There's a way to get them to form if you use an INK4A deleted background or P16 knockout mice. The problem is those mice are very, very prone to dying of other reasons. And so they're not practical to use. Eric Holland, who describes this model originally, many years ago, showed that you can get tumors to form the EGF expression, but it's difficult. The first question, yeah, again, great question. I as a surgeon, I'm always interested in supramaximal resection. I think there's value to it. I think Hughes Defoe has shown nicely that that it does help decrease tumor recurrence and progression. Function, you don't have a marker really. They don't take up some of these new kind of fancy tools like Gliomas, for example. So I tend to resect back to functional cortex depending where the tumor is. That tends to be my boundary. I do a lot of these surgeries awake just for that purpose, to make sure that we're not, but I like to be aggressive with these, because these are younger patients too, right? They're typically in their 30s and 40s, and you want, your best shot is their first surgery. So I think that's a reasonable philosophy now. And I think most people that do this surgery are kind of gravitating towards that posture.
I see. Thank you.
Dr. Rao, this is Eric Kuhlman. I'm also a neuro-oncologist. I have a couple of questions. On the FLP2 system, how did you administer the antibody to the mouse? And was there any side effect from the treatment?
IV, there are no, it's FGL2, there's no side effects to it in the mice that we saw. One of the challenges, though, of, we're obviously very excited about taking this to the next level. And my collaborator, Shulin Lee, has a company basically where he's trying to develop this. The problem is that there's high expression of this in the bone marrow. And so it's going to be a real challenge to figure out how to give it. Now, we didn't see any toxicity in the mice, but we're sort of trying to navigate this next step. So when we look at a bunch of human tissues and stain them, there's high expression in marrow. Again, is it navigatable? Probably, but it's going to be challenging.
And my other question is, what do you think about, you mentioned, you told this story about surviving, and it was a great talk, by the way, every segment was great. What do you think about surviving vaccine or CAR-T?
Yeah, so Survivin, it's been interesting. I didn't go into this in enough detail. Survivin, the original studies by Chakrabarty from Ohio State, I guess, years ago, showed Survivin. It's a fascinating gene. It's only expressed in the adult brain in tumor. You don't see it in normal brain. And so it was thought to be a home run for GBM treatment. And so Survivin vaccines were already developed, went through phase 1 and phase 2 trials, and haven't shown efficacy. And I think part of it may be this, that maybe it's a transcript variant. I don't know. Maybe they're targeting the common variant. It's not going to have an effect. For CAR-T, it probably does make some sense. A lot of challenges with CAR-T, as you know, for brain. But I think that it's probably one of those, it's so unique in terms of its restricted expression to the tumor and not in the normal brain, that it may be something that should be explored.
Great, thank you.
Sure.
Well, Ganesh, I just want to thank you for really a fantastic run through and exposing our group to the work you're doing. I know Raphael's extremely appreciative, as am I. And we look forward to future interactions, quite frankly. I think that your two link, maybe Texas and Boston aren't that far apart,
Right. Well, Chris, thanks so much for having me. I really appreciated talking to you all, and great to see you guys. And hopefully someday soon in the future, we can meet in person.
Thanks very much.
My pleasure.
Thank you. Thank you. Bye.